Now let's understand how DNA sequencing was done in Human Genomic Project. So first of all, let's understand what is genome. Genome is one full complement of genetic material. So when we are talking about human genome, it has 3 into 10 raised to 9 base pairs. It was a huge genome which had to be sequenced. And what is sequencing? To determine the order of these 4 bases, adenine, thymine, cytosine and guanine. So that was the DNA sequencing. That was the main purpose. And what was the purpose? Why they had to be done, the sequencing? To find out the basic composition of human genome, that what is the composition, and to find out the variations, that wherever there is a difference in the bases, and then the mutations, diseases related to that, to find out the evolution, that from if somebody is from Europe, what will be their sequences, and if somebody is from Asia, what will be their sequences. So to study all that, DNA sequencing had to be done. So for that, it was automated DNA sequencer that was used. Now this sequencer worked on the principle which was developed by this scientist Frederick Sanger. Frederick Sanger had also developed the same method for determining the amino acid sequences in protein. So he had found out the amino acid sequences, and that is why the same sequencer was used to find out the sequencing of the DNA. Now let's understand how did they do. Because the human genome was very huge, it had 3 into 10 raised to 9 base pairs, and this sequencer could only find out the sequences about 500 base pairs. So what they had to do is, they had to cut this DNA molecule into fragments, so that if the piece is smaller, then the sequencing can be done. So let's consider a small segment of DNA. We know that DNA molecule is very big, and even the fragment is quite big. Now let's understand how sequencing was done. So if we consider that this is a particular segment of DNA which has to be sequenced and then aligned, so for that, various enzymes were used to cut the DNA molecule. So first of all, if I consider this blue line, that is the one kind of enzyme which is cutting DNA at this point, then one of the fragments is the first one, that is marked in blue, ATTCGG. This is the first one. Now if we use another kind of enzyme which is marked in red, which is cutting the DNA fragment at a different point, and if we just read the middle part, that is GGTCAGG, which I have written in red. And then the third kind of enzyme was used, which is marked in yellow, cutting at this point. So it is AGGGCCAT. So first of all, different kind of enzymes were used in different media, and then the sequencing was done. After that, the overlapping regions were recognized. So overlapping regions is that when we see that in this fragment there is GG and here also there is GG. The last end has AGG and here also AGG. On the basis of these overlapping regions, the entire DNA molecule was sequenced. That this is the arrangement, that is starting from ATT and ending at CAT. So this is how sequencing was done, but it was not possible humanly to align and sequence the different fragments and then the entire genome. So that is why specialized computer-based programs were developed, and these sequences were then annotated and then assigned to every chromosome. So those computer programs which were developed to do the sequencing, because it was a mega project handling 3 into 10 raised to 9 base pairs, that was a part of field which was developed, and that field was bioinformatics. The last chromosome which was actually sequenced was chromosome 1, and it was done last in May 2006, including all other chromosomes, including even X and Y chromosome, all were sequenced earlier. Only chromosome 1 was the last one which was sequenced at the end in May 2006. So I hope you understood this. If you like my videos, then do subscribe to my channel and save and follow for more.